This presentation will be about Lab 8 in regards to Antibody IgG ELISA.
To preface this, 5 classes of immunoglobulins exist in humans: IgA, IgD, IgE, IgM, and IgG. IgG is the most abundant immunoglobulin, secreted by plasma cells and equally distributed in blood and tissue. The IgG class can be further divided into 4 subclasses. Each subclass varies in function and is produced in response to different Th subsets. The structure of immunoglobulin is made up of 2 heavy chains and 2 light chains that are linked together by disulfide bonds, weighing about 150 kilodaltons altogether. This results in the 4 polypeptide chains creating a tetrameric coordinated structure that forms 2 halves, which create a Y-like structure together. With 2 regions aiding in antibody-dependent cellular cytotoxicity, one region is the variable region that binds with high specificity to an antigen. The carboxy terminal constant region of the structure is involved in complement binding, placental passage, and binding to cellular membranes. The IgG class is involved in responding to foreign antigens, as it's capable of binding to extracellular pathogens. IgG can induce neutralization or opsonization for clearance of the pathogen via the complement system. To depict opsonization, all IgG variants can bind to Fc receptors and enhance phagocytosis by macrophages. As plasma cells must be formed, IgG presence signifies a mature B cell response, where B cells must be activated and class switching must occur. In this lab, the use of sandwich ELISA is appropriate for detecting the presence and concentration of our analyte, total IgG, in previously obtained serum samples from mice. Dilution of the sera is needed to curb the high concentration of IgG present in the samples. Similar to flow cytometry, our analyte here needs to be bound by an antibody. The assay involves trapping the analyte between a capture antibody and a detection antibody conjugated with an enzyme. The enzyme used here is HRP, which reacts with a substrate called TMB. The reaction between HRP and TMB results in the oxidation of TMB, which produces a blue color change. A higher level of detection antibody being present in the sample implies a higher level of oxidation occurring. This color change can be quantified in optical density units and correlates to the amount of analyte present in the sample, thus depicting the resulting IgG levels.
Prior to this lab, mice were inoculated with Streptococcus pneumoniae, an extracellular bacteria that induces humoral adaptive immune response, in which it involves antibodies. This pathogen is a common inhabitant of the respiratory tract of the host. Pneumonia is a leading cause of serious illness and death worldwide and is responsible for community-acquired infection of the lungs. Antibiotic treatment for infections typically includes broad-spectrum antibiotics. 4 samples were tested, 1 naive and 3 inoculated serum. 3 tubes per sample, 1 to 10, 1 to 1,000, and 1 to 10,000. 1 times diluent was used to make serial dilutions of mouse serum samples. For 1 to 10 dilution, 180 microliter was added to 20 microliter to tube 1 and was mixed. For the 1 to 1000 dilution, 495 microliter diluent was added with 5 microliter 1 to 10 dilution into tube 2. For the 1 to 10,000 dilution, 180 microliter of diluent was added with 20 microliter of 1 to 1000 dilution into tube 3. 50 microliter of 1 to 1000 was added to well number 1, and 50 microliter of 1 to 10,000 dilution was added to well 2. Each sample was added into pre-designated wells and were incubated at room temperature and in the dark for an hour. Following incubation for 4 times, wash buffer was added and contents were shaken out. The enzyme antibody conjugate was added into each well and then incubated at room temperature in the dark for half an hour. After incubation, the wells were washed and blotted 4 times again. TMB substrate solution was added into each well and was incubated in the dark at room temperature for approximately 10 minutes. Stop solution was then added into each well and the absorbance of the content in each well was determined. Here are our results. The standard curve for IgG was fitted with a trend line. The polynomial trend line has an R-square value of 0.9987, which indicates that this is a good fitting trend line. The concentration of IgG in both the inoculated and naive mice were measured at dilutions of 1 to 1,000 and 1 to 10,000. The concentration of IgG in the 1 to 1,000 sample for the naive mouse was 379.106 nanograms per milliliter.
The average concentration of IgG in the 1 to 1,000 sample for the inoculated mice was 307.272 nanograms per milliliter. The concentration of IgG in the 1 to 10,000 sample for the naive mouse was 38.312 nanograms per milliliter, and the average concentration of IgG into the 1 to 10,000 sample for the inoculated mice was 50.55 nanograms per milliliter. In dealing with mice inoculated with Streptococcus pneumoniae, it was expected to see a large T-cell dependent humoral response. In the lymph nodes, B cells BCRs can encounter antigens presented by APCs or free-floating antigens and receive signaling from the helper T cells. This will then allow for the generation of plasma cells and production of antibodies like IgG. Our results are inconclusive about the extent of the B cell response. The ELISA functioned properly, but the concentrations of IgG were not what was expected. The concentration of IgG in our Streptococcus pneumoniae inoculated mice was roughly the same as our naive. This value should be much higher, considering IgG is capable of enhancing phagocytosis, which is necessary to clear a pathogen like Streptococcus pneumoniae. The likely reason for the low level of IgG is that the mice were not infected for long enough. Somatic hypermutation allows for the generation of high-affinity antibodies, but this takes time. According to the CDC, the first time someone is exposed to a foreign substance, like a virus or a bacterium, it may take the immune system up to 2 weeks to make an antibody blueprint and to produce enough of a specific antibody to fight the infection. I recommend that if this lab is repeated next semester, that the infection manifests for at least 2 weeks in order to receive accurate IgG level results.